Welcome again to another Wiki Review for PABoardReview.org. My name is Wesley Norwood. I've created this Wiki Review on the physiology of blood pressure regulation. I will then tie this information into Joe's lecture on cardiac pharmacology. These are meant to be quick and enhance our understanding so that we can understand why and not just brutally memorize facts. When we're measuring a blood pressure, we are measuring the force exerted on the walls of the arteries. This gives us an idea of what's happening at the capillary or tissue level. Is the pressure adequate to deliver oxygen, nutrients, and carry away waste? Ultimately, an adequate and sustained blood pressure is what allows us to maintain consciousness, perform vital organ functions, etc., etc., etc. So when we look at blood pressure, we're looking at the sum of cardiac output times the peripheral resistance. The body maintains the blood pressure at a set point, and any deviation is regulated through both a short and a long-term mechanisms. Short-term blood pressure regulation is maintained by changing either the blood vessel diameter or the vascular tone, the heart rate, or the force of which the heart contracts, and it can do it all at once. But if you try to think of everything all at once, you're only going to confuse yourself. So you change one variable at a time. Long-term blood pressure is regulated by adjustment in the volume of blood in the system. Now we're going to look at everything at a very high level. You can go into more detail once you understand and have the basics down. So when the blood pressure needs to be adjusted quickly, it's via a neurological response or regulation. Remember, both heart rate and contractility are influenced both intrinsically and extrinsically, meaning inside the heart and outside the heart. A few examples of intrinsic regulation are the sinoatrial node pacemaker or the availability of calcium within the cardiac muscle. An example of extrinsic regulation would be neural stimulation or inhibitory signals sent by the autonomic nervous system or hormones. Keep in mind the cardioregulatory center is located in the medulla, and it both receives and sends inputs for blood pressure control. So receiving input. So there's baroreceptors located in both the carotid sinuses and the aortic arch. They send afferent or stimulations that's from afar via the glossopharyngeal nerve, cranial nerve number 9, and the vagus nerve, cranial nerve number 10, to the cardioregulatory center located in the medulla. I use the mnemonic coming from afar to remember afferent is traveling centrally. The vagus nerve, which is also part of the parasympathetic division of the ANS or autonomic nervous system, can send cardioinhibitory signals to the sinoatrial node. Remember, inhibitory neurotransmitters are acetylcholine. Remember that also that the word vagus means wanderer, and both sends and receives stimulation, which makes with many different pathways. To remember the role that the parasympathetic division plays, I think park and sit, rest the heart. The sympathetic division of the autonomic nervous system can send cardiostimulatory signals via the sympathetic chain ganglia to the SA node, which stimulates the heart rate and contractility. Remember, the sympathetic chain ganglia nerve cell bodies are located close to and on either side of the spinal cord in long chains that run from the neck to the pelvic region, called sympathetic chain ganglia. The tone or the diameter of both the arterial and venous vasculature is also influenced via the sympathetic chain efferent pathways. This influences peripheral resistance. Effective cardiac muscle contractions requires both an action potential and the correct amount of calcium. Remember, smooth muscle, skeletal muscle, and cardiac muscle allows different amounts of calcium to be released into the cytoplasm. The combination of ATP calcium is what allows the actin-myosin cross bridges to interact. This is what causes movement. All three types of muscles require calcium. Keep this in the back of your mind for when we discuss the cardiac pharmacology. Here's the overall slide with the short-term neurological regulations we just reviewed. Long-term blood pressure regulation is regulated via the renin-angiotensin-aldosterone system. Angiotensinogen is produced by the liver and released into the circulation. When a decrease in blood pressure is detected by the kidneys, an enzyme, renin, is released into the circulation. Renin then converts angiotensinogen to angiotensin 1. Angiotensin 1 will pass through the heart into the pulmonary circulation, where angiotensin converting enzymes, or ACE enzymes, located on the endothelial cells lining the pulmonary vasculature, converts angiotensin 1 to angiotensin 2. Cough can be a potential side effect in the use of ACE inhibitors, or angiotensin converting enzyme inhibitors. It makes sense why, because we're targeting enzymes in the pulmonary vasculature. ARBs are not associated with cough. More on this in the pharmacology section of the Wiki Review. Angiotensin 2 will circulate in the bloodstream and has a direct effect on the vascular smooth muscle. It causes vasoconstriction. This will cause the total peripheral resistance to increase, which will increase blood pressure. Angiotensin 2 also acts on the adrenal glands, causing the release of aldosterone. Aldosterone will retain sodium, water will follow sodium, potassium will be excreted. Because water is being retained, blood volume will go up, and so will the blood pressure. Keep aldosterone in mind when we're talking about the pharmacology part of this Wikipedia. Here we have the overall slide with the long-term blood pressure control via the renin-angiotensin-aldosterone system. To see the full video lecture, join us at pabordreview.org.